Hey everybody, my name is Adam. I am an MCAT tutor and top MCAT scorer here at Shemmassian Academic Consulting, and today I'm going to be walking you guys through a biology and biochemistry passage. So to start out, let's talk a little bit about the MCAT, right? So MCAT stands for Medical College Admissions Test, and it's a common test that applicants have to take in order to be admitted to most medical school programs that would be like MDs or DOs, and some other graduate programs ask for it as well. So it's a helpful test to be able to learn to do really well at, and if you're working on that right now to gain admission to one of these programs, hopefully this video can help. So where would you find a passage like this on the MCAT? So this is the biology and biochemistry section, which is the third section of the MCAT, right? So first section, chemistry and physics, right? You get 95 questions, or sorry, 59 questions in 95 minutes, then a short break. Then you have the CARS section, which would be critical analysis and reasoning section, where they just give you passages, you read them and analyze them, and you answer questions just based off of the passage. So it's basically just reading and answering questions. Then you get a slightly longer lunch break. Then this is the third section, that's where we are today, called the biology and biochemistry passage, where you have, again, 95 minutes to answer 59 questions. Then you get another short break, and then you finish out the exam with a 95-minute section on psychology and sociology, right? So 59 questions there as well, and then you're done. Total time, 6 hours and 15 minutes with breaks in between, so it ends up being around 7 hours of total time sitting, but 6 hours and 15 minutes of actual testing time. So in the biology and biochemistry section, when you get to that, there's a couple different strategies that you can use to manage like the way that you approach it, so you can give each question kind of your due diligence, or each question like the appropriate amount of effort. The basic strategy to start out with is there's 10 passages, and then there's 15 questions that are not based on a passage. We call those discrete questions. And an easy way to break it down is those first 15 questions, you skip to them, the ones that are not based on a passage, the discrete questions, and you answer those in the first 15 minutes, right? So 15 questions in 15 minutes. That takes 15 minutes off of our 95 down to 80 minutes. So now we have 10 passages to do in 80 minutes. The easiest way to break that down is to aim for 8 minutes a passage. It can be a little difficult to micromanage, like making sure you're getting exact 8 minutes per passage. So it might be helpful to break it up into like 3, 3, 3, and then 1, right? So you do like 24 minutes, and then 48 minutes, and then 72 minutes, and then 80, right? So you kind of like break it up that way, that you get a little bit of flexibility to go maybe to 9 or 10 minutes, or to even go a little quicker, maybe 7 minutes or 6 minutes within that like kind of 3 passage range. Because each passage is a little bit different in terms of difficulty, and in terms of length, and in terms of how many questions. We're going to do that for this passage. We're going to do kind of the basic starter strategy, where you read the passage and highlight what's important, and then you go over to the questions and you answer them based off of what you glean from the passage and what is outside information that we know the AAMC wants us to have to answer questions like these. We're not going to do this passage in 8 minutes together, because that is more of something you want to do on your own time. Instead, I'm going to be breaking this all down and explaining why I'm highlighting, what I'm getting as I'm reading, and then as I get to the questions, I'm going to be breaking down my full thought process as to how to really rule out every answer choice and pick the correct one. But for your own practice, you want to go quite a bit faster than this. Okay, so let's dive into it here. So I've got the passage here. It's biology, biochemistry, and it's 6 questions. So let's start by reading. The C subunit of ATP synthase forms part of the F0 complex and is embedded in the inner mitochondrial membrane. Right, so these are things that we're familiar with, right? ATP synthase, we're thinking that's at the end of our electron transport chain in the mitochondria, right? And they're mentioning the inner mitochondrial membrane, right? And that's where we create that protein gradient in which protons are pumped across that ATP synthase to create ATP, right? That's kind of our main, that's why we call it the mitochondria the powerhouse of the cell, right, because of this. So this is all outside knowledge that is coming to the forefront of our mind as we read these words. We'll see how much of it we have to use, but that's what they're kind of cluing us towards, the passage writers are cluing us towards with these words. Let's go on there. In its native conformation, the 75 residue long peptide folds into alpha helices and forms oligomers spanning the lipid bilayer. Right, so remember, lipid bilayer is going to be that membrane. Native conformation just means like what it looks like normally, like what it would normally form on its own. 75 residue would mean 75 amino acids, right? So amino acids can also be called residues, right? And amino acids is maybe the highest yield thing on the entire MCAT, so we should be familiar with all 20 of those. Right, peptide would be just like the linear chain, and then its folding would be like we're going into its secondary, tertiary, and then quaternary conformation to eventually form into our native. So alpha helices, that would be our secondary conformation, right? So like primary would just be the peptide. This would be a secondary structure. Okay, it goes across the lipid bilayer, right? We kind of knew that. So let's continue on here. Calcium-induced permeability transition, PT. Okay, so we're going to highlight this because anytime we see an acronym, we want to be able to know what that means. So in case we see it later and we forget, we can refer to it quickly. Occurs as a result of increased mitochondrial membrane permeability. So we'll call that, because we got to highlight this here so we can know what IMM, it's intermitochondrial membrane. Permeability by opening pores in response to toxic levels of reactive oxygen species and or calcium, right? So we should know what reactive oxygen species are, right? We have different things, right, that can lead to cancer, damage. We want to stabilize these with antioxidants, right? So, and we have ways of doing that within the body. We'll see what they ask about that. As a result, PT is believed to be a major contributor to cell damage under acute metabolic stress. That makes a lot of sense. So let's connect this conclusion here. So PT equals cell damage, or it's a big part of it. That makes a lot of sense, right? We make it more permeable. We have more reactive oxygen species then getting in and doing damage. All right, so the C subunit of the ATP synthase alone is sufficient to induce PT, right? So that's not good. Although the molecular basis of its participation is unknown. Okay, so we got to figure out how it does this. So we're talking about the C subunit, which we kind of already knew that, but just for this particular paragraph, we're going to note that that's what this paragraph is about. To investigate, I'm going to highlight the word investigate, because now we see this in almost every passage for biology and biochemistry, and even chemistry and physics section as well. But we often are doing some type of study to evaluate the statements that they made earlier. Like, so we made statements earlier in the passage, and now we're doing some type of a study or investigation here. So to investigate the possibility of its direct participation in PT through forming an ion-conducting pore, the peptide was solubilized in a detergent solution and inserted into model lipid bilayers. Okay, so we're just doing a study. Let's see what it does. The peptide was incubated in a buffer designed to mimic a membrane-like environment. Okay, the resulting far UV circular dichronism, right, so far UV CD spectrum suggested the formation of beta sheets and was very different from the CD spectrum of the C unit under native, right? So now that we see that we've got another secondary and that's also very different, we remember earlier that they mentioned that it was an alpha helix natively, right? So now it became beta, right? That was formed, beta sheets were formed. That's very interesting. That could be why. Very different from its native. Right, I'm going to highlight the word native. The protein structure was further analyzed using predictive modeling. The results are shown in figure 1. Okay, so so look, we see it came under the next line, but this is figure 1 here, right? And as we zoom in on this a little bit, we can kind of see what we got going here. When we see figures, we want to just take note of what are our axes, what are our trends, is there anything significant to note from what they were talking about, right? So we got residue number, which remember that's how many amino acids we have on the X, and we got score, right? So score is, I guess, likelihood. or something along those lines. They didn't really specify here. Let's look. If we don't know, we can come over here. So we have predicted secondary structure of the C subunit based on probability calculation. So I guess this would be like probability. So I'm guessing as we go up in score, we're more likely to be that particular conformation. So right, remember we had, this was 75 residues, right, as we go up to this first paragraph. So that's slightly off the end here, but right, we might have different numbers here. Just interesting to note, we got a beta peak here, while alpha goes very low, almost down to 0.8, the alpha helix. And then for the most part, they kind of go opposite of each other, right? At about 60, beta goes down, alpha goes up. So yeah, interesting to just note. There's not really a whole lot going on there. But yeah, that's going to be our predicted secondary structure. Next, the C subunit in beta sheet conformation was tested. Okay, so this is a new test. So we're going to highlight the word next to know that we're doing a new thing now. The formation of cross-beta aggregates using a THT fluorescent reporter was observed. Okay, so again, still beta. So I'll highlight beta again. The aggregation process appeared to be strongly suppressed by 1 millimolar calcium solution. Okay, so we were able to stop it with this. Okay, the channel open state conductance of the C subunit channels alone and in the presence of calcium was also quantified. The results are shown in figure 2. Okay, so we got no calcium, 302 PS, which I guess that would be just a certain unit, right? It looks like it's conductance, PS, so how much conductance? So a little bit less conductance with 302 and more with 1 millimole, which I guess that's kind of what they said here. We were able to strongly suppress the aggregation process, which means we were still able to keep that kind of that membrane difference, right, with that potential difference there with calcium. So kind of just agreeing with that statement there. All right, then we go on. As a result, the C subunit was concluded to be capable of self-assembling into beta sheet oligomers. Okay, so this is the conclusion, right? Result, concluded, self-assemble into beta sheet, and that its observed ionophoretic properties are linked to its amyloidogenic nature. Amyloidogenic nature. Okay, cool, so that's the conclusion. So again, that's a normal passage that you'd often see. A lot of the stuff in this particular passage is beyond the scope of the MCAT. And right, how you're going to be able to figure that out is the more passages that you practice and the more familiar you get with the content that's on the MCAT, the more you'll be able to recognize, okay, a lot of this stuff is not fair game, but it's related to things that is outside knowledge, right? So to give you a little example of some of the outside knowledge that they were kind of alluding to here, we're going to go over to my second page here where they talked about our secondary structures, right? Alpha helices and beta pleated sheets from our MCAT content guides on Shemmassian.com. Yeah, there's some really helpful content guides. Here's a couple pictures from it. But right, this is our protein folding, right? When we're taking that primary structure of all of our residues of amino acids, and then they're going to be folded into certain conformations. So the first kind that we can have is called an alpha helix here. And it's going to look kind of like a spring like this over here on the left. And then here's what it would actually look like. And if I draw it, if we were looking down from the top like this, right, if we had our alpha helix kind of curling around like this, and then we're looking down from the top, if you notice, we can look over here as well. All of our R's, which would be like our side chains of our amino acids, are facing in the middle. We have an occasional one that comes out like this, but the majority of them are kind of internal, right? The majority of them are kind of going to be in like this, like they're going to be R's in this way. And why is that, right? It's because for alpha helices, a lot of our amino acids, not all, but most, are going to be non-polar, or we can call that lipophilic, right? We have our 4 different groups of amino acids, right? We got our polar, our non-polar, and then we have our charged and positive and negative charged amino acids. Those are our 4 main groups. So alpha helixes are going to have primarily nonpolar amino acids because they're going to usually span across a membrane where we need to be nonpolar, right? So an alpha helix, and we saw that here, this is a transmembrane protein as an alpha helix. And then they also mentioned beta-pleated sheets, right? So over here on the right, we got 2 different versions of beta-pleated sheets, right? Again, this is still a secondary conformation. One other note before I get into the beta-pleated sheet, when we're thinking about primary, secondary, tertiary, and then finally quaternary conformations, those are all different types of interactions between our amino acids, right? So primary is literally just end to end, right? It's just going to be N to C, right? N terminus to C terminus. We're not going to have any interactions between side chains or even between non-adjacent amino acids. Nothing yet, right? It's just primary. And then our secondary interactions is now we're making interactions between non-adjacent backbones. So we call this backbone interactions, right? So a backbone interaction means like where our C terminus, right? We have a carboxylic acid over here, right? And then like our N terminus has a hydrogen attached to it, right? If we had a non-adjacent one kind of over here with our double bonded oxygen, like that up here, we can actually form a hydrogen bond there between our hydrogen and our oxygen, right? Because hydrogen bonds can form between nitrogen, oxygen, right, and a few other of those highly electronegative elements, right? So we can form a hydrogen bond between non-adjacent amino acids. That would be considered a backbone interaction, right? Not using those side chains. That's going to be secondary. And then tertiary, right, what's now added is now we can do side chains, right? Our side chains can now interact within each other. That's where we can have like our disulfide linkages, right? Our really strong covalent linkage, or even a hydrogen bonding, or our positive negative ionic interactions between our positively charged amino acids and our negatively charged amino acids, right? That's all side chain interactions. That's going to be tertiary. And then quaternary is, now that we have those side chains interacted, quaternary is now our subunits, right? Identical or different subunits, right? We can have like, say like a homodimer, that would mean 2 identical subunits, or like a heterotrimer, which would be 3 different subunits, right? That's going to be what we would consider quaternary conformations, right? So that's just a little small detail into protein folding and kind of how we form our proteins from just individual amino acids, right? And how we kind of break this up, at least for the MCAT, as primary, secondary, tertiary, and quaternary, right? So if they're trying to figure out what type of conformation are we in, primary, secondary, tertiary, or quaternary, we want to look for this main thing, right? Just the straight up chain, backbone interactions, and then side chain interactions, and then finally interactions between subunits. Okay, so now let's look at this beta-pleated sheet, right? Again, this is still secondary interaction, right? So we're just seeing backbone interactions, right? And we have parallel and anti-parallel, right? Those are our 2 different conformations that we can make. And right, so parallel, everything is going to be running in the same direction. You can kind of see how this comes around to the top, right? I have a bonding like that, so it's all going to be going down, and then again going down again, or vice versa, right? Up and then back to up, right? And we still have those backbone interactions, right? You can see them in the middle here. You can see these hydrogen bonds occurring between our hydrogen and our oxygen, right? From our nitrogen to our oxygen like that. And here again in this spot. Right, so that's why we know this is a secondary interaction, and it's going to form like an actual sheet that looks like this, right? And then anti-parallel is the same thing, except for it's going to go down and then back up, and then right again back down and then up. So we're going to kind of alternate between up and down in terms of direction from N to C. So yeah, and one of the other kind of important things to note about beta-pleated sheets is that we have this thing at the end here called a turn, right? A turn, and there's going to be 2 main amino acids that are used for a turn, right? And it has to do with being able to actually make that tight turn, and that's going to be proline and glycine, right? They're 2 nonpolar amino acids, but the reason why this works is proline is our most rigid amino acid, right? It's really, really tight. You can't really move our angle of how much it creates a turn, so it naturally is going to start our amino acid on our turn. And then glycine is our most flexible amino acid, which means it can then correct us back to going straight. So if you imagine if we had a proline here, right, and it's really tight and we're getting really tight like this, then if we have a glycine here, it is so flexible that it can actually make us go back straight to be able to connect and form this hydrogen bond here and then form the next one as well because we had proline glycine. Right, so proline glycine is going to be our two key amino acids to note that we're probably making a turn in a beta-depleted sheet. And then alpha helices want to look primarily for non-polar amino acids because that's typically their function within a protein. Okay, so that's a little bit of background information from the MCAT. So let's dive into these questions now. Okay, so our first question, we got amyloid proteins are directly implicated in the pathogenesis of which of the following? Okay, so this is kind of we call this a pseudo-discrete question. And when it comes to MCAT biology and biochemistry questions, sometimes our questions are all based on the passage, and sometimes they look a little bit like this is just telling us, okay, we mentioned an amyloid protein in the passage, but this is all outside information. It's pseudo-discrete. You could answer this question without reading the passage, which, it's kind of nice. We don't have to use any of our passage knowledge to answer this, but then it's also kind of not nice because you need to have the outside knowledge. So it's kind of like a double-edged sword there. So let's see if we can answer it here. So amyloid proteins are pathogenesis, right? So like some kind of disease of which of the following, right? So Alzheimer's, right? We remember that beta amyloid plaques are often present in the brain for Alzheimer's disease, so that one's going to be true. Okay, and so let's look down at our answer choices. So 3 of the 4 of them have 1 in them, so we can only rule out B at this point. And then let's look prions, right? So prions are actually kind of like a sub-classification of amyloid proteins, right? And they're going to be involved in pathogenesis of different types of infections and stuff like that, right? So prions is a yes as well. So we already have our answer, right? Our answer is going to be C because Alzheimer's and prions are both, and we know that they can't be 1, 2, and 3. It's not an option. But just for exercise sake, let's look at 3. So 3 is Parkinson's disease, right? And the association that should come to mind when you see that is dopamine, right? So Parkinson's disease is a disease of dopamine, right? Our dopaminergic neurons are not functioning right, so we don't have enough dopamine in our brain, right, causing like those tremors and stuff that you typically see of Parkinson's disease, because dopamine helps us to stabilize and be able to like use those muscles in a way to keep our hands still instead of shaking and having that Parkinson's tremors, right? So 3, that has nothing to do with amyloid proteins. That has mostly to do with dopamine and dopaminergic neurons. So good. So got number 1. Let's move on to number 2 here. Which of the following amino acids is most likely to be found at the N-terminus of the peptide? Okay, remember, so we're just talking about the peptide itself, right? So if we go back over, they said this a couple times, but in the native conformation, it's an alpha helix, right? And then again down here where it said that it turned into a beta sheet, that's very different from the native, which if beta sheet is different, then it must have been alpha helix. So twice they mentioned that it was probably an alpha helix. So what's most likely at the N-terminus, right? Remember what we talked about from our content guides is we're looking for a non-polar amino acid, right? So we got phenylalanine, which is both aromatic and very nonpolar, so that's a good one. And then B, we got proline, that's also nonpolar. C, we have serine, that's going to be polar, so that's out. And then aspartate, right, that's going to be polar and charged, right? So C and D are out, but we're definitely between A and B here. So how do we choose between 2 nonpolar, right? Remember what we also talked about. Proline, even though it's nonpolar, it's actually going to be a good marker for a beta-depleted sheet, right? Because if we see proline, we're thinking we're probably going to be making that turn. So proline is one of the few amino acids, nonpolar amino acids, that wouldn't be present in an alpha helix because it can't do that kind of just fine turning that we do kind of in spinning forming that alpha helix, right? Because it's too tight of a turn, more likely to be used in a beta pleated sheet. So definitely going to be phenylalanine here. Okay, number 3, we got inner mitochondrial membrane permeability may be slowed or stopped by the action of, right? So if we kind of go back over and look at what we were talking about with IMM, right, it's going to be this paragraph, right? So IMM is kind of due to PT, permeability transition, in response to toxic levels of reactive oxygen species or calcium, right? So what can we do? We know we can stabilize reactive oxygen species, or maybe like produce or secrete calcium. We're going to look for something along that lines. We want to reactive oxygen species and or calcium is what we're going to look for in our answer choices. That's how we impact our membrane permeability. So number 3 here, inner mitochondrial membrane may be slowed or stopped by the action of, right, electron transport chain, right? All we're really using for that is NADH, FADH2, and we're producing protons to like create that proton gradient that then the ATP synthase uses. So neither of those have to do with calcium or reactive oxygen species. So A is not going to be a good answer. We're gonna cross that one out. B, pentose phosphate pathway, right? We're going to form a couple different things, right? NADPH is one of them, and then ribose 5-phosphate, right? So that we can produce more of those precursors to DNA and RNA, right? Ribose is going to be part of RNA, right? And then deoxyribose is going to be a part of DNA. So we need ribose 5-phosphate for both of those. But NADPH, right, might be helpful for us here, right? Because NADPH, it's not the same thing as NADH, right? NADPH is going to be used in our glutathione pathway, right? And we should remember glutathione because glutathione is one of our main molecules in our body that we use to stabilize free radicals or reactive oxygen species. We call that an antioxidant. So pentose phosphate pathway is directly connected to creating NADPH, which is used to produce usable glutathione from its oxidation state to its reduced state where it can go and stabilize free radicals. So B is a good one because it's talking about that first one, right? The free radicals or the reactive oxygen species. Okay, and then C is the citric acid cycle, right? Again, what are our products of the citric acid cycle? It's going to be NADPH mostly, some GTP, right? That's not going to be good. We want calcium, reactive oxygen species stabilization. Neither of those are good. And then glycogenesis, right? We're producing glycogen, and we may even be using ATP for that, right? Not related to calcium or reactive oxygen species. So B is the only one that relates, and it very directly relates, right? NADPH directly catalyzes that glutathione pathway creation. So good. That's a good answer there. A little bit tough, lower yield content knowledge question there, but we should be familiar with all of these different pathways and kind of what they're used for. So definitely fair game. All right, number 4 here. The wavelength of light used to produce figure 1 falls somewhere on the range of, okay, right, so let's go over and check to see what kind of light we used. So we used far UV light, right? So we should be able to recall, for the most part, there's at least 2 numbers that we absolutely know we have to recall, and that's the ends of our visible light spectrum, right? And those numbers are 350 to 750, right? So 350 is going to be our highest energy, and then 750 is going to be our lowest energy. And if you can't remember, right, so you know, everybody knows the rainbow, right? Roy G. Biv, right, with violet on the end and R being red, and those are all the colors in between. But what's hard for people to remember is which end is high energy and which end is low energy, right? And so the way that I remember it is, right, we have ultraviolet on one end and infrared on the other end, and I'm like, okay, which one is going to cause damage to my skin? What do I wear sunblock for? I want to block those UV rays because it's going to be damaging, so those are going to be the high energy. So that's how I remember that violet is the high energy or the low wavelength, and red is the low energy or the longer wavelengths, right? The longer wavelength side. So, right, 350 to 750 is our visible range, right? So 550 to 600 is going to be somewhere in that visible range, kind of in the middle, maybe in the orange, yellow, green, kind of maybe between yellow, green, or in yellow or in green, kind of in that middle range somewhere in there. And then B, 200 to 250, right, that's going to be slightly smaller in terms of wavelength, which means higher in energy, which could be pretty close, right? It's right underneath 350. This could easily be UV. We'll see if there's a better answer, but for right now we're leaning B. C, 850 to 900, right, that's going to be longer than red, right, because 750 is red. So it's outside of visible, it's probably infrared. And then D, 0.01 to 10, that is very small. So that's, we're probably going past ultraviolet into even higher energy. And I think, okay, if ultraviolet causes damage to my skin, what goes deeper? X-rays would penetrate deeper. And then, if you have too many X-rays, you can actually cause damage to your body overall and even develop cancer, right? So it's even more damaging. So yeah, this is probably an X-ray. So it goes a little bit deeper than ultraviolet. So we're going to go with B here. That seems to be the best answer. Okay, and then number 5 here. The action of the C subunit in beta sheet conformation is most analogous to that of a ligand-gated ion channel, a voltage-gated ion, a porin, or an ABC transporter, right? That's going to be ATP binding, right? So let's see. What did we have going on here with the beta sheet? I think, let's see. It says the channel open state conductance, right? So calcium is just kind of traveling through. We don't really have any bonding. Let's see, is there anything earlier on when we talked about it going into a beta sheet? It looks like an ion conducting pore. It looks like it's just a pore that ions are traveling through, right? Ion conducting pore. We don't have binding to it in a bilayer. So we probably don't want to lean towards ligand-gated, right? Because ligand-gated would mean we need a ligand to bind to it, and then it opens. Voltage-gated, we need a change in voltage, which, if anything, this ion channel seems to be working when our membrane becomes more permeable, right? Which means we would have even less voltage, if any, at all. So A and B are not good answers. We can X both of those out. Pour-in is more along the lines of what we're looking for. It just opens and lets things through, right, under certain conditions. So C is probably pretty good, and then we don't have any ATP going on anywhere in the passage. The only thing we talk about is calcium and reactive oxygen and different circumstances in which that happens, right? Nothing to do with ATP, right? So not going to be that. All right, so last question here. Based on the information presented in the passage, which of the following statements is most likely to be true? Okay, so we got chaperone proteins required to produce functional F0 complex. That's possible, right? Because we have to transition from an alpha helix to, or from a beta pleated sheet to an alpha helix. So in order to make that, we might need chaperone proteins. The chemiosmotic gradient is required to produce functional F0 complex. Probably not. It almost seems like we need to have a functional F0 complex in order to create the chemiosmotic gradients. Like, this is kind of the wrong way around. See, the F1 complex of ATPase does not play a role in permeability transition. Maybe the statement could be true, but right, the passage didn't talk about that F1 complex. It's all about the F0 and the C subunit of the F0. So unlikely. And then the F1 complex, again, we're talking about F1. We could keep reading if we want, but it might be true, might not be true. We don't really know. The passage didn't talk about it. So right, we can rule C and D out because we don't know. That's not fair game. And right, we said B was kind of the opposite of what the passage was talking about. So then E, therefore, must be the correct answer, or A, sorry, must be the correct answer. Chaperone proteins are required. That makes sense, right? We were trying to make a functional F0 complex. We needed our C subunit to be in a certain spot. So A, requiring a chaperone protein would be totally possible there. Okay, so there we go. That's our 6 questions there. I hope that was helpful for you guys. I hope that you guys agree with me, but I feel pretty good about those. Those were pretty reasonable questions. So yeah, if you enjoyed this, give the video a like, or if this was helpful for you. And then in our description below, we have a little link that you can click that you can get a question of the day sent to your inbox every single day. It's just going to be an MCAT question. And that can help you keep up your studying. So yeah, I hope you guys have a good one, and happy studying, everybody.